physicians. It's an incredible space we're in, and we're very, very grateful to be here. And of course, huge thanks for everyone for being here. It's a very, very exciting time for Neuro-Bio as a company. There's quite a lot to talk about, and I'm sure, as you'll hear from our amazing panelists throughout the morning, the idea that Alzheimer's is anywhere near even chipped away at as a problem it's really not the case. The fact that we've been trying to find a solution and a treatment for so long as a medical field, as a society, without understanding how to even measure and diagnose the problem initially, means that we've been flying completely blind. We've been shooting into the dark and not knowing if anything landed. Part of the story of Neuro-Bio is, of course, the therapeutic, but the other side is very much the diagnostic. We need to understand whether or not we're moving the needle. We need to understand as a scientific field, both for ourselves and for the wider society in the field, that what is working? Why is it working? Let's explain the underlying fundamental mechanisms and really get to grips as to why Alzheimer's happens, why this is such a big problem, why do certain parts of the population are predisposed to this. So I won't take any real time this morning to sort of talk about any of my views, which I'm sure we'll talk about later. But I just wanted to really underline the fact that this is going to be a little bit more of a bilateral discussion. Yes, you'll hear from our amazing panelists on their views, but you'll have very much an opportunity to not just go through a Q&A with us afterwards, but put your own thoughts forward, challenge some of the views that you'll hear. This is an amazing opportunity for everyone around the room to have a bilateral discussion. This is not going to be a lecture theatre of university. It's an idea that we've tested, we have data on, but very much we don't want to be prescriptive. We want to be very much proactive with everyone here. So without further ado, and you'll have to excuse me, I'm going against my key rule of anything, which is not to have notes in front of me. But unfortunately, with the CVs like our lovely panel have, which in length genuinely do rival Tolstoy's first running draft of War and Peace in length and impressive nature, I'll break that one rule that I have. So you'll be hearing from Dr. Neus Falgàs, who is a neurologist and researcher at the Alzheimer's unit of the Hospital Clinic de Barcelona. She specialized in the diagnostic assessment of cognitive disorders and the clinical management of those diseases. Her overarching goal of the research that she does is to improve the diagnostic framework of Alzheimer's disease. So we've been working very closely with her clinical team, and we very much look forward to working even further. You'll be hearing from Dr. David Bunker, who is the Chief Technical Officer of Aptamer Group, who, again, very recently we've partnered with on the diagnostic space. He holds a PhD in molecular biology and has spent nearly 20 years, I won't say exactly how many, to protect your ego, developing nucleic acid aptamers. And aptamers, for us, are a huge area of opportunity in the diagnostic space, and we're very, very excited to be chipping away at that. You'll be hearing from a director of Neuro-Bio, Professor Paul Herrling, who has held the corporate research head position at Novartis. He's been the chairman of the board of the Novartis Institute of Tropical Diseases in Singapore. He is the Professor Emeritus of Drug Discovery Sciences at the University of Basel. So keeping a foot on the academic and the corporate side, which is always very, very interesting to talk about. And he's a trustee of the Foundation for the National Institutes for Health, where he is also the chairman of the Biomarker consortium. So again, he'll have very, very interesting views on what you'll be hearing shortly. And you'll be hearing from immediately now, of course, the Baroness Susan Greenfield, who is the CEO and founder of Neuro-Bio. She's, I mean, again, I don't need to tell anyone, but she is one of the world's leading voices in neuroscience, one of the most recognized faces and names in the field. She holds over 200 peer-reviewed papers to her name. I don't even know where to start with everything else. The first woman ever to give the Royal Institution Christmas lectures, and she then went on to become the director of the Royal Institution herself. She holds 32 honorary degrees, awards such as the Royal Society, Michael Faraday Prize. She's an honorary fellow of the Royal College of Physicians, which is where we find ourselves today. And of course, as well as being a key patron of the Alzheimer's Research UK charity. So without further ado, I'll pass on to people much better positioned to take the seat. Thank you.
Thank you very much, Mike. Well, we all know why we're here. We're here primarily to celebrate 10 years of Neuro-Bio. And this reminds me a bit, I don't know if when you went to kids' parties, you had to have the sandwiches before you had the cake. I don't know if anyone remembers that. This is a bit like the sandwiches, the sort of serious bit, before we have full celebrations for most of us tomorrow. But thank you all for coming at this early hour. It's wonderful to see such a mixture of sectors, and I'm aware that some people are clinicians, some are science and specialists, others are very dear old friends. So forgive me if I address this in very general terms. Some of you know the story already. Some of you might be thinking, oh, why doesn't she sit down? I want to hear the rest of the panel because we've heard what she has to say. I feel I do have to, nonetheless, give a bit of background because it's important that we're all on the same page before you hear from my co-panelists, whom I'd like to thank very much for coming. All of them from afar, from York, from Barcelona, and from Switzerland. So it's great that we're all together. And I think, as Mike was mentioning, having these different approaches will give us perhaps some exciting new ideas in the otherwise very depressing landscape of the unmet clinical need that is Alzheimer's. Okay, so just to remind you who we are. We're based at Culham. And you can see here the lovely labs. Many of you have seen the labs, but some of you haven't. And this was thanks to our Australian shareholders who let us refurbish the labs, which is quite unusual for a biotech, that we didn't have to spend it on direct cost. We could actually refurbish the lab. We were founded 10 years ago, obviously. We now have, it's increasing almost by the minute. I spoke to Anton, who's here yesterday, our lawyer, and it's now 20 patent families we have, isn't it? At least 20. At least 20, yeah. Nearly 20, yeah. Over 19 patent families. This encompasses 6 independent assets, and the team, all of whom are here with one exception, Samuel, who is holding the fort back at base. We have 4 postdoc scientists, 6 junior scientists, and 9 management, and more of them tomorrow. Okay, so why has it been so long to develop a treatment for Alzheimer's? Well, putting my head above the parapet, my own view, perhaps shared by some, not others, is there's too much focus on the amyloid drugs. And we've had a tsunami of publicity on the 2 amyloid antibodies that have, just before Christmas and just recently, grabbed all the headlines. Just to remind you, amyloid is an abnormal deposit found in the brain outside of the cells that has been combated by 2 drugs, donanemab, and this latest one, from lecanemab and donanemab. I can't pronounce them always because the complications of the antibodies at the end. They have met with limited success. It might be the others will talk about this, or it might come up in the discussion. But I have always said that this isn't really the be-all and end-all, that amyloid, of course, is a factor, but it's a downstream factor. And this is articulated in this review I did about a year ago, which was published. The other reason that it's been so long to develop a treatment for Alzheimer's and that still bedevils it is that neuronal populations have distinct profiles, rather like these gentlemen in ill-fitting suits. One size doesn't fit all. And most theories will assume that. Most theories will talk about abnormalities or problems at the level of the single cell. We argue, no, you should look at neuronal populations which have their own neurochemical and physiological signatures. And it might be that we see differences in how the different brain areas react. Because we know neurodegeneration is a highly selective process. It only occurs initially, as you'll hear from Dr. Falgàs, in one cell group at the beginning. And so if we knew why these cells were particularly vulnerable, then we could target them specifically, we could understand how they worked, and perhaps treat before we have the decline in the cognitive symptoms. So we need to know how the vulnerable cells differ. That's the first question that Neuro-Bio has asked, but to the best of my knowledge, very few others have actually asked this. Well, people have asked it, but not many people have been listening to them. So back in 1981, Martin Rosser had this visionary editorial highlighting an area of the brain he called the isodendritic core, which sounds a bit of a mouthful. The isodendritic core, meaning equal and characterized by very vast arborization of dendrites in a sort of tangle. Dendrites are the sort of branches that come out of cells. And he highlighted these cells shown in white as the particularly vulnerable cells that are the first to degenerate in Alzheimer's and Parkinson's disease. This idea, although it didn't get huge traction, nonetheless has been carried on, and there's this, to my mind, very good review which summarizes this in 2015 that was written, and again here, where the interesting feature is that although these cells are the primarily vulnerable ones that go, you can see here the so-called BRAC staging, levels 1 to 6, shown on the left on the y-axis, are actually bereft of amyloid at the early stages. So that would suggest that although amyloid is a very legitimate downstream marker to target, it can't be the primary cause. How can it be when, if in the early stages, in the primarily vulnerable cells, it doesn't really exist? So we need to think about something else. What do they have in common? Do they have a common nuclear, a chemical profile? Is there something they have in common? What they have in common is how they originated at 4 weeks gestation, where they come from the basal plate of the cell. This is what you look like when you were 4 weeks in your mother's womb. It's the basal plate that is the primary origin of these cells, not the alar plate. Why is that interesting or important? Well, you have these light bulb moments sometimes, and with the luxury of being at that time at a university and teaching and having time to reflect and join up dots, I remember the moment I read this review back in '96, where Nancy Wolfe, the author, had actually classified these different cell groups according to their embryological provenance. And what was fascinating, what she called the global systems, which are in fact the isodendritic cells, are actually the ones that are the primary cell groups to degenerate, the Raphe-nucleus, Leucocereus, and so on. So what's really interesting is we need to explore how they're different. And as you can see, one of the other things that they have, shown in the red circle, is they exhibit a robust plasticity that is not there in their alar-derived counterparts. Now, could that be one of the reasons, the fact that they are still sensitive to growth factors that could make them particularly vulnerable to the cells? I'm going to summarize now, so people that are expert, please forgive me, I'm going to show a very little film that takes this idea forward. So let's play the movie.
Here at Neuro-Bio, there are two interrelated projects about which we are very excited. First of all, the prospect of a revolutionary blood test for Alzheimer's, which could show that an individual's brain had already started to degenerate, and as such that they would go on to develop the dreaded symptoms of Alzheimer's, memory loss and confusion, 10 or 20 years later. Complementing that test, we envisage a therapeutic which could be administered via a nasal spray to stabilise cell loss. Even if a patient had already received a diagnosis, the cognitive impairment at the very least wouldn't get any worse. However, if administered earlier, before the appearance of symptoms, the symptoms would never appear. We consider that this would be the most effective approach to treating Alzheimer's to date. This video will show how the therapeutic works in a way the non-specialist can understand. So let's start at the beginning. At 4 weeks gestation, there are already two different layers of cells in the developing brain. Firstly, the alar plate, where the majority of neurons come from. Secondly, the basal plate, which then constitutes the vulnerable neurons, forming a kind of hub. The hub neurons have the distinct property of retaining the ability for regrowth. They're able to do this by mobilizing a chemical messenger that we've identified, T14. Neurodegeneration will be triggered if these vulnerable hub cells are in any way damaged and T14 activated. This is because T14 is a Jekyll and Hyde molecule. In development, it promotes growth, but in the mature brain, the context has changed, so the same process is now toxic. As T14 continues to increase in this area, the damage eventually progresses to the rest of the brain. This results in Alzheimer's disease. Here you can see the difference between a healthy brain and a brain with advanced Alzheimer's. The brain has less volume and shrinks as T14 continues to damage and kill cells. Neuro-Bio has devised a highly novel therapeutic drug that stops T14. It's called NBP14. Our goal is that NBP14, when given intranasally, reaches the brain and acts on the vulnerable neurons. The drug displaces T14 from complexing with its target and thus stops further damage to cells. The result of developing a blood test which could detect the T14 means that eventually we could offer a routine blood test, rather like for checking cholesterol. Then, before any symptoms arise, prescribe the NBP14 and prevent the onset of cognitive impairment that characterises Alzheimer's disease. Thanks for watching. I hope you're as excited about this as we are.
Okay, so I won't do a question to test who knows what NBP14 is now, or T14. So you've been introduced, therefore, to T14, which I used to call, until Professor Ziri stopped me calling it the evil molecule. But she said, oh, you have to call it evil. Do you remember Margaret? Yes. But I think of it, and it's something comes off, that it's an agent that promotes growth, but can turn to be toxic in maturity. But let's look at the proof that it promotes growth. This is recent work with George Mason University, Nadine Kabani's lab, and she has shown in tissue cultures, if you look at the control, compared to peptide-treated cells, there is a clear extension in the lampopodia that the cells have grown. In our own work from Hippocampus a while ago, you can compare the controls with the T14 treated, where, as the red arrow helpfully indicates, you can see the sort of enhanced neurites that resulted from treatment with the peptide. So far, so good. But it can turn toxic if you increase the amount. So you've just seen the low levels and how you've got the extended neurites, the Jekyll situation, the Hyde situation. You can see the cells are in much poorer shape when you increase the dose. Similarly, you can increase the application and show that for a short application it enhances growth. If you increase it, then it is toxic. But really, what we find very interesting is not so much going along this trophic toxic axis, as it were, according to amount or duration, but to age. And others have shown, Emeritus and Schramm have shown, back in the 90s, that another crucial factor determining whether an agent promoting growth is going to turn toxic or not is actually the age of the cells. So in a developing system, one can imagine the T14 will indeed be beneficial, but in more mature brains, it would, the same amount for the same time would now be toxic for reasons we can go into if you want to get technical later. So this is what we can see happen, that in the youngest brain, this is this is human brain, it's highest. This is work from Joanna, unpublished in the lab. You can see here in adults, nonetheless, there is an age-related decline in normal aging, as would befit something that is normally going to promote cell growth. That is the same in the developing rat, but in the Alzheimer brain, you can see it recapitulates development. It's almost doubled in the midbrain as part of the isodendritic core. So that shows you that this theory does hold water, that neurodegeneration could be an inappropriate form of development driven by T14, inappropriately activated in the context of a mature brain. So to summarize, and from Neus Falgàs, I've adapted this figure. She might have a similar one, but I'd like Neus to have credit for this figure because she has the nice pictures of the brain sections. I had the idea of the traffic lights, so I don't know if you've got the traffic lights. Okay, so in the normal brain, back stage 0, there's T14 present, but it's not doing very much. Everyone is healthy, T14 levels are low, you don't have any other problems, you're not depressed, well, you might be, but not really, and you're sleeping fine. Now, say there was damage, as saying in the film, it could be anything. It could be free radical attack due to declining scavenging mechanisms. It could be a blow to the head. It could be, doesn't really matter. It could be toxins. It doesn't matter what triggers it. But if these cells are damaged, bear in mind they have retained the ability to grow again, unlike the other cells in the brain. Because they've retained the ability to grow again, what they will do is recruit T14 to try and compensate for that. So the T14 will go up. But remember, this is now an adult brain, and so therefore you're going to have problems. Now, what I think Dr. Falgàs will talk about more is the interesting relationship that perhaps people haven't mentioned much of changes in mood and sleep disorders, which would fit with problems with these cell groups, which are very basic cell groups in the brain that underlie arousal and biorhythms. Nonetheless, the T14 presses on. Because these are very basic primitive cells, your memory is just fine. Because there's a high degree of redundancy with them, we know that. They're functioning to all outward purposes, seems fine, but the problem is carrying on. The T14 is carrying on to promote through a feed-forward system, more cells dying, more cells go on. Eventually, the damage will spread transsynaptically to the more sophisticated parts of the brain, such as the hippocampus and the cortex. You will then present with memory problems. That's where you'll see the amyloid as the main feature, and that's when action is taken. So you can see that you are missing out on that amber period, which really we want to capitalize on, and that's what we really want to address now as we're talking. So T14 is upstream of the traditional markers. So I want to stress, yes, we do acknowledge amyloid plays its part, but it plays its part later on. And you can see that T14 or T30, which is its sort of cousin, same molecular idea, that they do cause an increase in amyloid and tau, both in tissue cultures, in brain slices, and indeed in animal models of Alzheimer's, the most typical one being the so-called 5-Fab model, 5 times familial Alzheimer's, where in the transgene negative animals you see very little T14, but in the model you can see clearly there's T14 reactive plaques, showing that again it's closely associated with a pathology. So that shows you T14 as a feature of the pathological brain. And we've done early work with Nottingham University and Evotec showing that if you introduce the peptide into healthy rodents, the neurons go down. When you treat with our bioactive peptide, T15 is a control agent, and you can see that does nothing. Amyloid beta goes up and tau goes up. So clearly there is work here to be done, and that's what we'd hope to do in the future for developing a non-genetic and much more authentic animal model that would replicate the full clinical profile of Alzheimer's. Oh, I don't know what's happened here. Oh, suddenly, what's happened? Okay, sorry. Someone's got bored of the... sorry, you're getting a... Can you sort that out? Sorry, it's just suddenly galloped on. I think someone just got a bit bored with the talk. Okay, so let's hope that now works. Fine. Okay, so how can it be blocked? Well, you've probably seen the preview, that's MBP14. MBP14 will displace, it's a bit like a person with their arms open. But if we can tie the hands behind their back, as shown here in so-called cyclation, then it will displace the toxic agent. Therefore, it can't do anything because it's cyclated, hands tied behind its back, and the naturally occurring agent can't do anything either. So that's how it works. And this was quite novel. So when you administer this to the animal model mice, the genetic animal model, they actually have an improved weight gain that is more apparent as time goes on, as you can see in the red. With something called novel object recognition shown here, where they have to show if they remember something or not. And if something is novel for a rodent, they'll explore it more. And so therefore you can see just how long they take to explore something as a test of their memory. If they explore it more, it means that they see it, they think it's novel. And you can see here in normal wild-type animals, the red shows the higher exploration. They've remembered what object is novel. Well, you can't remember a novel object. They recognize an object as novel. In the Alzheimer model, treated with saline, however, that is not the case. They can't distinguish between novel and familiar, whereas in a comparable group of animals, animal model, treated twice a week for 14 weeks, with MBP14, you can see their memory is as good as the normal mice. And moreover, if you look at amyloid in the brain, you can see there is a significant drop in amyloid. In fact, it stops the production of amyloid altogether in these animals. So as you can see, this heartened us very much for the therapy. We now have, going forward to so-called IND, investigation of new drug, a company called Bachem, based in Switzerland, who are making it at pure grade and with a high yield. A company called Evotec are looking at the safety and toxicology, which we've only just started, but so far it's looking very promising. It's detectable in the blood, it hangs around, there's no adverse effects in 3 times the dose given. Still a lot to do there. And we've been officially recognized by the MHRA, our equivalent of the FDA, as it being a truly, truly novel drug. Anton did these patents for us, which we're very excited about. I don't know if the numbers have changed, Anton, since you last gave, but the anti-amyloid drugs, there's 2,500 patents, anti-tau 894, and anti-T14 there's about 6, and we own them all. So those numbers might be changing, Anton, I don't know, but you can see we're in, I hope, a very comfortable position. This has been recognized. We're finally getting traction because this is such a novel idea by The Lancet, and I'm delighted that all but one of the authors of The Lancet article that we're trying to put forward. No, we don't know if it's out yet, but it was commissioned and it's been submitted to the Lancet. And these are the 4 authors, I say, Neus, Falgàs, and Margaret Azaria here. We're hoping Mary Sally will come tomorrow. Sadly, Sharon's in the States, so it was hard of her to come. But it shows that we're getting recognition, and we're all on tenterhooks to see what happens with that. The final reason about its being so long is this long time lag, and that's what we're really talking about today. We disagree with Bill Gates, shown here, and that we think it's not, we don't want to do it before the symptoms get too bad. We want to do it before they even appear. This is a very well-known kind of picture where you can see MCI stands for mild cognitive impairment. You can see there's a lost time window. Now, Brack staging, as you can see, the first 2 stages traditionally are without symptoms. And up until now, to the best of my knowledge, there's been no marker that will reflect that it's already started. But last year, we were able to identify T14 increasing at the pre-symptomatic stage. And this got us very excited, because if this happens in the brain, all we have to do now is reproduce that in some kind of peripheral system that can be accessed in living people. And then we would have a first ever pre-symptomatic marker that would help enormously, as we'll probably hear later, for what that would do. So what we're doing so far is we're looking at saliva. We are trying to get, in combination with advisors on regulation, analysis done by the Rosalind Franklin Institute, and I'm delighted to announce today that we've just had an award from the Rosalind Franklin Institute that enables us to now go forward, and that's the work of Anna Ashton, who's here, and Sarah Herrling. I'd like to congratulate them on the application that they did. That will go forward in the autumn, and then with the help of GNL, what we hope to do is submit to the FDA a pack based on some preliminary samples that would then be ready to go to a full clinical trial. However, what you really need to do is you need to be able to screen something yourself or screen something without having to go to mass spectrometry. What you really need is a biomarker, let's say lateral flow test, or what Americans call an antigen test, because what we'd really like to do is have something available for a very preliminary early screen before you do all the fancy, highly quantitative stuff, which would follow on later. You want a go, no-go kind of test. And then eventually, if the person was referred to medical authorities and then they decided there was a problem, you'd start on the medication, ideally our MBP14. And if that, as we hope, stops any symptoms developing, it means you'd never get the symptoms if you carried on taking the drug. That's our dream. So where are we with the biomarker? What I'd like to show you now is something, I'm going to end on this because it's hot off the press. This was done by Joanna with samples from Neus. So it's a very timely moment to unveil this. And we've, thanks to Anton now, made it. I can safely reveal it. So we know that in CSF, 50% of nasal secretions are derived from CSF. So anything you see in CSF, you could quite legitimately extrapolate to perhaps you could then look in lateral flow in nasal secretions. And what Joanna found, which astonished us, was in healthy controls in their CSF, an extraordinary high correlation, despite the small n, of phosphorylated tau, which is the cardinal intracellular marker, and the first one that characterizes the pathology, with T14, with a 0.0008 significance level. So a 0.95 correlation for biology for an N of 7 is really astonishing, and particularly interesting when you compare it with a comparable number of samples from mild cognitive impairment, where it does not exist at all. So this suggests that what you could devise, and this is what we're thinking of, is looking at the ratio of p-tau to T14, because unlike looking at absolute levels of something, let's say like tau, you wouldn't be dependent on grouping it with ages or saying this is age match or it has to be under certain medications, because the person could effectively be their own control. They'd be their own benchmark, which would make it, I think, a much more accurate metric, a single metric to look at, for simply screening, is someone healthy? If they are deviating from this metric, then they get referred. That's all. Nothing, you don't do any diagnosis. It's simply a screen, which, as we may hear later, I would imagine at the very least would be helpful for recruiting people for clinical trials, because you could eliminate people that were going to be less likely to benefit from your drug. So that is my final slide. It's one that we are very excited about. Yes, I know the ends are small and all the rest of it, but I think you'll agree that the association of T14 with p-tau is something that really is further vindication of this idea. And let's hope that we can harness that to develop not just a first-in-class therapy that actually works, but a marker that predicts you need that therapy before you get the symptoms. Thank you very much.
Good morning, and thank you very much for attending our meeting. Thank you for the introduction. So Susan has given you a summary of the growing body of scientific evidence that implicates T14 in the deterioration of the brain, and how one could make a drug that might stop this, or at least reduce the speed of degeneration. What I would like to do is to focus a little bit on the importance of T14 also as a biomarker. And in order to do that, one needs to understand a little bit of the history of why everybody was running after APP for so long. And so it starts, and this historical review, I'll do it short, I can do it under 3 hours, don't worry. Don't worry. So what we, no, you don't have to go, it's not going to be that. So it starts with Alzheimer, of course, with Alois Alzheimer. Around 1900, he was a neurologist and a medical doctor, and he observed elderly patients who had this typical cognitive deteriorations in his patient group. At that time, obviously, we didn't have molecular biology. We didn't have an MRI. The fashion, the scientific fashion at that time was slicing brains and coloring them with dyes. And that's what the great discovery of Golgi and Ramon y Cajal and these people made, and Alois Alzheimer. And his discovery was that he could see in slices of a brain of such patient, which has a typical cognitive deterioration, he could see what he did not see in other patients, or not many, roundish structure that he called plaques. And they were in varying numbers, but he made it a hallmark of Alzheimer's disease. So all the people who died with this, with these symptoms, were sliced. Now, if you think about it, however, it's all called plaques. But the reason it's called plaques is because these were horizontal slides or slices, but they're slices, thin slices. But independent of the plane of cutting, you always saw this roundish thing, which means they have to be spheres and not plaques. So you find these spheres with some strange material which he could color, but he could not define what it was. Later, it took actually many years until molecular biology progressed to the point where it was shown that in these plaques you have a protein, the amyloid precursor protein, which now we know sits in the membranes and has various functions. So people were interested in APP. For many years, nobody really understood the function of APP. We just knew that it was there and in those spheres. So the hypothesis that this APP in those spheres was very important and might be causal for the disease was very much strengthened even later when genetics, molecular genetics improved, and when families were found, some in Sardinia, some in the north. In these families, you had a very high incidence of Alzheimer, and the hallmark was that this Alzheimer started not in the 80s as it does in the sporadic case, which is the majority, 90% of the Alzheimer case are the sporadic case. But in these families, the patients showed the symptoms around 40 and 50. So people went down into the genome of these families and tried to find mutations that might correlate with it. And they found in the APP gene itself, which is in those spheres, they found mutations. And those mutations they could show was increasing the level of APP. So you can understand that that was a very powerful argument in favor of the APP hypothesis. And it is, so now, today we're doubting the APP hypothesis as a causal agent, but it was absolutely clear that at that time, as a scientist, you had to test the hypothesis because it was by far the best available and the evidence was pretty powerful. So that's what people did, including myself when I was head of neuroscience at Sandoz at the time. We had transgenic mice with these mutations and we studied on there. And so people tried now to get at APP and testing the hypothesis that it was causal. The easiest thing is let's get rid of it. Let's try to get it away from the brain and see if things improve. And that's where the people started to develop antibodies. Antibodies that could bind to the APP, to this protein, and then by normal natural mechanism that antibodies get eliminated, try to get rid of the APP. And it took another, a huge amount of years until such drugs were developed. They were specific enough for APP. And of course, many of the critics said, oh, well, antibodies will never get into the brain and you're not going to do anything. But first it was found that APP can be seen in CSF, so outside the brain, and even in the blood sometimes. And you also had increasing data that shows that in Alzheimer, the blood-brain barrier is weaker than it would be in a normal person. So they did the experiment, first in animals, and you can see you can completely clear those transgenic animals from all APP in their brain. They got approval to go to man, so they did that. And they started to see that you can actually reduce the level of APP in the brain of human beings with the symptoms of Alzheimer. So, however, these drugs did, or these antibodies did what they should do. But the results, in clinical results, in terms of improving of cognition, were very, very minor, if at all. Actually, several drugs just failed in phase 3 because you couldn't demonstrate any effect. And that was obviously major disappointment to the whole field. And there, I will just point out 3 hypotheses why that might be the case. One is APP, as many people had also discussed and suspected but didn't know, might be a side effect kind of the neurodegeneration, but not the cause. And so even if you get rid of all the APP, the cause is still there somewhere. Now, one thing you should also say is that in these studies, they're not as benign as people thought at the beginning because they're antibodies. You could see serious side effects. One, the brain would shrink in these patients that were treated with the antibodies. And that is easily understandable because there is so much APP in those patients that when you take it out, well, the volume is lost. That would not be a big problem. However, you also had blood leakage. Some studies showed that many of these plaques sit around microcapillaries, and when the APP goes away, it leaks. That's a much less benign side effect. So, and the third one is a statistical argument. Is as Susan had pointed out before, it was difficult to distinguish real Alzheimer patients from all the other neurodegenerations that occur in age. So people are just treating all the people, let's say, I don't know, beyond 80 or 75, that had cognitive deterioration. But that collects people with APP and some without, and only later was people starting to try to measure APP itself. But when I say statistical, so in the samples of many of those studies, you had people with cognitive deterioration, which was the common point, but some had APP, some had no APP, some had, and so you dilute your sample, and you can't show a statistical effect in your clinical studies. It's all, so these are three hypotheses why. There's one that I didn't mention, is that actually that also has to do with regulatory issues. That is, the FDA or the MHRA will only give you permission to test an experimental drug, a new one, in patients that might get benefit from this drug, because there is a risk of it with a new drug. And so they allowed people to test the new drugs, these antibodies, only in patients that showed very distinct, clear deterioration of their minds. And so the argument by many people is to say, well, by the time that's the case, so much of the brain is destroyed that there's no way you can repair it. It's just too late. And that started actually a whole school of thinking, and maybe Neus will talk about it, which is to try to test these antibodies, but much earlier. And the dilemma comes at the point where Susan mentioned, is, fine, and how to see that? And now you start to see the importance of T14. So first, let me see my notes here. Good. So the advantage of notes is that they don't run away with you like that. So what turns out then, if T14 is indeed part of this neurodegeneration process, and it is actually not inconsistent with everything that was described before. You saw the tau relationship, because tau was always recognized as also one factor in neurodegeneration. And this correlation is very, very important because it tells us that T14 is not something that comes out of the blue into the brain and destroys it, but it's consistent with the old or other hypotheses around Alzheimer. So now, of course, you can think of making a medicine to stop this. And the key, however, is to select the patients that have this neurodegenerative process, and not all of them, and dilute your sample. And that is why this last slide of Susan's was so very important. If it is true you can detect T14 during the whole life of a human being, starting in embryos, as we saw, I just have a little crazy hypothesis which you can shoot down later in the discussion about T14. As you know, when we all get born, we have many more neurons in our brains than we have now. It's not only age, but actually around birth and when the brain matures, there is a pruning process going on which selects those pathways in the brain and those neurons that are needed for survival. And the others are actually eliminated. So my crazy hypothesis is that T14 might be one of those factors that eliminates the unwanted. And that is why it is so present in very early age. And if it reappears late in life by some malfunction, which Susan described, then it might still prune, but then it prunes neurons that you actually really much would like to have. Now, I have to say, I have zero data on this. It's just pure speculation from history and what you know about brain and humans and evolution. And maybe somebody will take it up and show it. But anyway, the key is, and that is the importance of being able to measure T14 in patients with zero symptoms. Because you know, probability is that the neurodegenerative process will end up in cognition deficits that are a hallmark of Alzheimer. But now you have the tool in hand to be able to start treating, and especially with drugs like NBP14, which can block the effect, the negative effect of T14, you can start treating before visible degeneration of cognition occurs. And this would be a complete breakthrough in the field. But not only medically, it is obviously for the patients themselves, which can keep their cognitive independence much longer. They can live at home normally. They don't need, every Alzheimer patient in advanced stage needs 2 or 3 people to maintain them. It is key for society, because the cost of Alzheimer on my generation, which is the baby boomers, will now go into the age where Alzheimer numbers will go up like crazy, and you too. This cost to society will be humongous unless we can dampen that. And so an approach like was described today, with the tool of detecting early and the tool to be able to dampen the effect, the negative effect of T14, might completely change the outlook in this respect. So I think that is as far as I would like to share my ideas with you. And I think put in the context, you can realize what an absolutely important discovery this is. And of course, I also have to do the usual caveats. We still have a long way to go. We have to go through clinical studies to validate the biomarkers. And that's typically very hard. It was said I'm a member of the Biomarkers Consortium. People work for years and years to get a biomarker validated by the FDA, for instance. So it's hard work. But again, just as at the APP time, the problem of Alzheimer degeneration in our societies is so important in terms of medical need that it's absolutely worth doing everything we can to try to test this hypothesis, the T14 hypothesis. So I encourage you all to help. Thank you very much.
Thank you, everybody, for attending today, and thank you especially for the invitation. It's an honor to be in this panel. Should I change the slides myself? Okay, today I am talking a little bit about the underlying changes, pathology connected to the clinical symptoms and the importance of early diagnosis and treatment. So we classically understood Alzheimer's disease as a group of symptoms, which is memory loss, disorientations, etc. But on the last decades, we knew the hallmarks of the Alzheimer's disease. We were capable of detecting them by biomarkers during life. So we changed the conceptualization of the disease. We now know that it's a clinical biological entity that has a wide clinical phenotype, starting from what are called preclinical stages, but I don't really like this word. I will tell you later why. Preclinical stages in which there are some AD changes already happening in the brain, then some clinical symptoms in form of mild cognitive impairment, mild memory loss will take place, and on later stages we will finally have dementia. And we are right now capable to diagnose the disease from mild cognitive impairment stages. That much as we can do. And there are these two hallmarks of Alzheimer's disease. The first, the well-known amyloid, and then tau pathology and neurodegeneration. In terms of amyloid, I will describe a little bit the characteristics in opposition to tau pathology. Amyloid aggregates in the brain as amyloid plaques, extracellular plaques, and all of them are distributed predominantly within the cortex. The distribution of these plaques has nothing to do with the symptoms or the degeneration, the patterns of degeneration within the brain. So all the patients with Alzheimer's disease have a pretty similar distribution of amyloid plaques within the cortex. In the case of tau, the pattern is a little bit different. Tau aggregates in form of neurofibrillary tangles, and starts not in the cortex, but in subcortical areas of the brain, and later on it reaches the cortical areas. And the distribution, it's closely linked to the symptoms and the atrophy patterns. Here I have, can you hear me anyway? Without Here we have this image in which
there is a picture of MRI, which shows atrophy patterns, tau PET, which shows the tau distribution within the cortical areas, and amyloid PET that shows amyloid distribution. And we have three different cases as an example. The first is an amnestic presentation of Alzheimer's disease, which is predominantly memory problems. Then we have a visuospatial presentation in which patients have visual problems specifically, and it's a rare form of Alzheimer's disease, but still it's Alzheimer's disease. And in the last example, we have a language variant. We can see that, I don't think it's working. It's okay. In the case of amyloid PET, you can see that the distribution is similar in these three cases, but in the case of tau PET, in the case of the amnestic presentation, tau is distributed in the medial temporal cortex, so that's the area of memory, and it makes sense because memory impairment, and tau is restricted into memory areas. In the case of visuospatial presentation, the tau is deposited into the occipital and parietal areas, which are connected to these visual spatial deficits. And in the last example, the language presentation, tau is distributed into the language areas, the temporal area of the brain. So confirming that tau is related to the phenotype, to the clinical symptoms, and where the brain starts to degenerate first. And this has been confirmed not only in vivo with imaging, but also in neuropathological studies. And this is a nice work of UCSF in which they detected that the amnestic syndrome had a lot of tau on hippocampus, and other phenotypes had this kind of distribution of tau. So in the temporal areas, in the language variants, and in the occipital areas, predominantly in the visuospatial syndrome. So the conclusion is that tau drives specific patterns of neurodegeneration and the clinical symptoms in AD in opposition to amyloid. And we, in terms of neuropathology, have this description of the amyloid pathology and tau pathology staging. We know that amyloid starts in the cortex, and it only reaches subcortical areas at very, very late in the disease stages. And tau pathology, back in the first stage in 1991, Brack described that hippocampus was the first area of the brain affected by tau. And it makes sense because hippocampus is linked to memory problems. But in 2011, Brack stage changed. He had to review the staging because he realized, and others that did a lot of research in this area, that tau pathology starts within the brainstem, within the subcortical areas, before it reaches the hippocampus. So the first neuropathological AD changes that we see happen in the brainstem due to tau pathology when amyloid is still not present in the cortical areas or anywhere really. And these subcortical areas are called the isodendritic core, which are a group, a network of different nuclei with different function. They release different neurotransmitters, the cholinesterase, the serotonin, orinergic, or the noradrenaline. And I will center my attention into noradrenergic system, which is this neurotransmitter is released by the locus coeruleus, which is a brainstem nuclei, that it's the main releaser of noradrenaline and has been more widely studied because tau pathology staging has highlighted this nuclei as the first seed for tau pathology. And these locus coeruleus neurons in Alzheimer's disease have been demonstrated to decrease its volume in neuropathological studies for each Brack stage. Specifically, the LC volume decreases about 8% of its volume. And this is not innocuous. Of course, it has some clinical implications. The degeneration of the LC nuclei and all the other isodendritic core nuclei make the development of some subtle symptoms that are not memory, but are sleep problems or depression or other mood alteration. In this study, they performed clinical pathological correlations, and they realized that before the tau pathology reached the hippocampal area, so when the tau pathology is restricted into the brainstem, the odds to develop sleep problems or depression or anxiety were higher, demonstrating that this link between tau degeneration of the subcortical nuclei and these non-memory symptoms, but still symptoms of Alzheimer's disease. There have been also many studies investigating specific patterns of sleep with brainstem axodendritic core degeneration in Alzheimer's disease, and not only in Alzheimer's disease, but other diseases. For instance, this study, they described 2 different patterns of sleep in Alzheimer's disease and another tauopathy, which is the paralysis supranuclear palsy. And they saw that the PSP had an hyperinsomnia and Alzheimer's disease had an hypersomnia during the day. And they realized that the degeneration patterns within the brainstem in the different areas were accordingly to these different clinical phenotypes. So the subcortical systems is a primary mechanism associated with the sleep disturbances in the early stages of neurodegenerative diseases, showing the PSP and Alzheimer's disease an opposite clinical pattern and also a opposition pattern of neurodegeneration. But not only clinical pathological correlations, but in vivo studies, not only in Alzheimer's, but in older adults, have shown that there is a relationship between these sleep symptoms and the degeneration of the LC. For instance, the higher the LC degeneration, measured by MRI, the higher the nocturnal awakenings in cognitively healthy adults, and the increase of plasma total tau levels. And other in vivo studies have put the attention in the mutation carriers to see whether the pattern could be similar. And LC degeneration was found to be associated with the age of onset of the disease in mutation carriers. And you can see the difference in the first graph with the ones that are cognitively healthy and the ones that have the mutation. And this LC degeneration was also linked to the tau pathology within the brain measured by PET tau and also to the cognitive symptoms. This is some of the data, preliminary data of our center, the hospital clinic, in which we are measuring the severity of neuropsychiatric symptom by the neuropsychiatric inventory and the volume of locus coeruleus by the neuromelanin sensitive MRI. And we observe, as you can see in the graph, that the locus coeruleus is decreasing along the AD stages, similarly of what we have seen in the neuropathology, and it is directly correlated with the severity of neuropsychiatric symptoms. So the lower the volume, the higher the neuropsychiatric symptoms. And this has implications in early stages because the point is that the LC tau degeneration, and not only LC but other subcortical nuclei in the brain, translate into clinical problems, into symptoms, in the similar way that it does in the cortical areas. So when a lot of patients that come to the clinic, you can, for a diagnosis of Alzheimer's disease, you can see that they are already have been prescribed antidepressants or sleep medications, because probably we are talking about a precognitive period of Alzheimer's disease in opposition to a preclinical period. And this happens in this wide time frame of 15, 20 years before the memory problems start. And this is already disease, although they are not the core symptoms of the disease, because we describe the disease based on memory problems, but really we can see prior symptoms that are sentinel before the memory problems start. And we have a precedent for this. For instance, the Parkinson's disease, or within the same spectrum of alpha-synuclein and the Lewy body disease, in which we understood many years ago, we have this conceptualization of the Parkinson's disease as a motor pure syndrome. And we realized many years later that it has a premotor phase in which we could observe sleep problems, such as the REM sleep behavior disorder, or mood alteration, such as depression, when the alpha-synuclein was confined into the brainstem. And later, when alpha-synuclein reaches cortical areas, we can the motor symptoms or even cognitive symptoms start to arise. In summary, what I wanted to highlight is that the AD physiopathology is complex and it goes beyond amyloid, and it has multiple actors, and the tau pathology is one of them, and also the subcortical areas. Tau pathology is involved in pre-amyloid stages, so is involved earlier in the disease course, within this preclinical phase of the disease, in which we have learned that it's not that preclinical, it's only precognitive. And when tau is confined to these subcortical areas, is driving these sentinel symptoms, and brain damage is already been establishing because tau is driving neurodegeneration within these areas. And of course, we are still figuring out what is the ultimate trigger for the AD and tau pathology, and the hypothesis of T14 offers for the first time a plausible explanation for that. So I think that taking into account all of the actors, not only amyloid, but tau and subcortical areas, is the formula to success in terms of finding an earlier diagnosis and treatment for Alzheimer's disease. And of course, it has diagnosis and treatment implications. Right now, we only have the symptomatic treatment for Alzheimer's disease, like memantine or others that are mildly effective. And there have been these anti-amyloid drugs that are, if finally approved everywhere, we will be able to give them to our patients once we diagnose in mild cognitive stages and dementia stages. But this is a bit too late because we will be able to remove somehow some proteins of Alzheimer's disease, amyloid and specific, and this may somehow slow disease progression, but these anti-amyloid therapies are not going to change. They will have no effect on all the established degeneration and all the established symptoms within these 15 or 20 years before. So, of course, it's nice to have new treatments, and I think that we are in a sweet moment in the Alzheimer's disease field because different treatments and approaches have been developed. But I think that the NBP14 is especially groundbreaking because if we are able to diagnose or measure at risk for Alzheimer's disease before the memory problems start, before these 15, 20 years before memory problems, we will be able to avoid neurodegeneration within subcortical and cortical areas of the brain, not only of amyloid, but tau, and we will be able to prevent the symptoms, not trying to just stop them where they are and try to slow it down, but prevent them. And these were the thoughts that I wanted to share. I will be happy to discuss or answer any questions.
Perfect, thank you. So first of all, thanks very much to Susan for inviting us down to be part of this very special occasion for, so it's a great pleasure to be here, thank you very much. So my name's Dave Bunker. I'm not a neuroscientist, so there will be no pictures of brains in my presentation whatsoever. But hopefully I'll be able to show you sort of how this project came to light, what we do as a company, and why I'm here today. So first off, I'm the Chief Technical Officer and one of the co-founders of Aptamer Group. So as a company, we develop novel affinity ligands called aptamers. So these are nucleic acid-based affinity ligands. Now, how this story all started is this building here. So just over 6 months ago now, Susan was kind enough to come and open our new facility. And as scientists do at events like this, we got chatting, and immediately we could see an opportunity. Susan had a target, we had a technology that we could put together and start to build something that we hope will form the basis of a diagnostic device for detection of Alzheimer's disease. So it's all started around this new building. So Aptamer Group are based up in York. We're a team of about 40 people, developing, as I mentioned before, nucleic acid-based affinity ligands called aptamers. So first off, for those who don't know what an affinity ligand is, it's basically a molecule that sticks to another molecule. But those affinity ligands are used absolutely everywhere in the life sciences. You cannot get away from affinity ligands. They're used in diagnostics, they're used in purification reagents, they're used in a lot of therapeutics. They are everywhere. And the one that most people are aware of are antibodies, shown as that purple molecule there. Now, antibodies have been around for a very long time. They're very good at their job. They do what they do really well. But they don't do everything. There is a real need for other molecules, and I'll come back to that in a minute. So just first of all, to show you the difference between an aptamer and an antibody. So you can see here a cartoon of an antibody. The binding region is just this very tip portion here. The rest of that molecule is essentially a scaffold. And there's not a lot of flexibility around that molecule. You've got a little bit of movement around that joint, a little bit of flexibility, but not a great deal else is going on. You can see, in comparison, the aptamer just next to it is about 1/10 the size, so a lot smaller, and importantly, it's a nucleic acid. It's not a protein. And there's a lot of advantages that come with that. First off, it's a very, very flexible little molecule. So what you can't quite make out in the schematic there, but you can, there's a small molecule inside that aptamer. The aptamer is wrapping around it, so it's almost caging that molecule. So the dynamics allow aptamers to interact with targets that other molecules like antibodies just can't get to. So just a little bit on the history of antibodies. So right over the far side, back in 1890 was when the first antibodies were discovered. Back then, they didn't really know what antibodies were. They just knew there was something in that case, it was actually in horse blood. There was actually something there that they could make use of. Okay, and back here in, where have we gone? Back in 1975, just at the bottom there, was when we first started to describe the processes for generating monoclonal antibodies. So you can see that was nearly 100 years later. About 10 years later than that, in 1986, was when the first monoclonal antibodies were actually entering the clinic. Okay, so about mid-80s. In the early 90s, other technologies started to emerge. Now, the reason for that is because people were starting to recognize the problems with antibodies. And I was actually at an event not a million miles away from here last Thursday talking about exactly that, the problem with antibodies. So it's well known in the literature. I mean, you've got some really great titled articles here, one about the antibody horror show. Basically, this whole event was around the fact that approximately 50% of antibodies fail to meet performance criteria. They don't do what they're needed to do. They cross-react, they're not specific enough, they're not selective enough, they're not high enough affinity. They don't really work very well. So there's a real need for alternative technologies like ours. So these nucleic acid-based affinity ligands have advantages because they're essentially synthetic. Okay? So there's no animals involved at any point in their development, and there's no animals or really biology involved at any stage in their manufacture. They are essentially chemicals, okay? Which means they're much easier to identify, they're much easier to control, and they're much easier to manufacture. So we can generate our binders against a much broader range of targets. So if you can imagine, if you have a toxic molecule that you put into a bunny rabbit, probably not going to get an antibody against that. You're going to end up with a dead bunny rabbit. So we can work with targets like that because our process doesn't rely on animals. Ours actually relies on robots, which are a lot cooler. Okay? We can also control a lot more of the process around the way the aptamers are isolated. So we can make sure we select them in the right sort of setting for their end application. So we get over some of these problems with antibodies not working in the right setting. And as we've heard earlier, some of those settings could be, for example, nasal secretions or CSF. I'll come back to that in a minute. As I mentioned, the selection process is entirely in vitro, so it's all on robots. But importantly, especially for projects like this, where we're going to turn this into a product, the advantages at the bottom. So the production is chemical. It's solid phase synthesis. So once you know what an aptamer is, you can make it again and again and again and again, and you can scale it up. You don't have the batch-to-batch reproducibility issues that you can have with antibodies. So I was chatting to a couple of the team from Neuro-Bio outside before the event started. You know, if you imagine a lateral flow test that maybe has a couple of micrograms in each test, if you can have an aptamer that you can synthesize on a kilogram scale and you use a microgram in that test, that's a lot of tests before you need to do any sort of revalidation. So batch-to-batch variability is much less of an issue because you need fewer batches. Okay? It's much easier to work with. You can also never lose it. Once you've got an aptamer, it's essentially information. It's a sequence. So my laptop in my bag, I probably shouldn't say it, but there's quite a lot of aptamers in that bag on my laptop. Okay? So it's sequence. You can't lose it. And one of the great things about the technology from a lateral flow point of view, or generally from test point of view, they can be stored dry. They're very soluble little molecules. So when you put them onto a lateral flow and dry that test strip out, you're going to have no problems with the stability. You can ship them all over the world at room temperature. You don't need cold chain. So there's a lot of advantages of using technologies like this in, for example, lateral flow. So as a company, we see ourselves as an enabler of technologies. We take customers' targets, they have something they want to work with, they have an application, something they want to do with it, but no way of making the two things marry up in a timely fashion. So, in the case of the event today, the target, T14, the end application, a diagnostic. How do we make those two things come together? An aptamer. One of the great things about this technology, and I'm sure Anton will be very grateful for this slide, the fact we can develop these things very, very quickly. So you might be surprised to hear that we actually kicked off our collaboration with the team at Neuro-Bio at the start of May, I think it was, and we submitted a patent on the aptamers that are going into this test yesterday. That's fast. Okay. So, a couple of examples of the sort of thing we can do, and hopefully you'll see where I'm going with this and how we get into where we are today. So we can generate our binders against all sorts of things. So, for example, we can look at them on the surface of cells. So you could have a biomarker that you want to go after, a cell surface receptor. You could just have a cell you want to go after. It could be a cancer cell, it could be a neuron. Doesn't really matter to us. We can select those aptamers against either that protein or that whole cell, or both. So a couple of examples of the sort of thing you might want to do with that. Well, if you've got a binder that recognizes a cell, obviously you can put a fluorophore on that and use it in flow cytometry. I'm not going to go through, there's lots of data here, don't worry about it. You could use it in flow cytometry, you can use it in imaging. You can also attach all sorts of drugs to it. So you could attach a nucleic acid-based drug, an siRNA or an ASO, and use it to deliver. So you could use an aptamer as a delivery vehicle. You could also attach other drug molecules to it, like a chemotherapeutic, and use it to take that drug where it needs to be. So as we said before, without affinity ligands, a lot of these tests, a lot of these drugs don't work. If you've got a drug, it needs to go where it needs to work, and an aptamer can help to do that. Other end of the scale, so we've got cells at the top. We can also work with small molecules. This is some work that we did with a group in Italy, generating binders against CPT11, a chemotherapeutic precursor. You can see here that the aptamer recognizes that CPT11, shown there in blue on the little biosensor trace. What it doesn't recognize in this case is SN38, which is the active drug metabolite. So what you can see there is how this aptamer could be used to monitor conversion of CPT11 into SN38. We actually have a companion aptamer that goes with that recognizes both molecules. So it's not beyond the realm of man to see how you could use one aptamer to say, right, I've got both of these molecules here and this other aptamer here to monitor conversion of one to another. So we can use these binders to look at conversion of drug molecule into either metabolite or prodrug or whatever. The great thing about this was the team actually did a comparative study using an aptamer-based biosensor that they developed using our aptamer and their gold standard, which was LCMS. And you can see there an absolutely lovely correlation between the LCMS results that they were getting and the aptamer results that we'd helped them to develop. And importantly, the aptamer was very specific. So it recognized CPT11, but didn't recognize any of these other medications that patients may already be on. So it was very, very specific. Now, getting a bit closer to what we're actually here, what we're talking about today, is developing binders against proteins. So you can see in a variety of tests the way that the aptamers could work here. So we could use them either as a sandwich pair, recognizing two different epitopes on a target, for example, T14. You could also use them in conjunction with an antibody. If you already have an antibody that's very reliable, you can have an aptamer antibody hybrid and make a pair that way. The point is, they work in this sort of assay regardless of what the other affinity ligand is. Now, what you can see on the data set on the right-hand side of the slide closest to me is essentially using the sensor platform here to develop binder pairs or characterize binder pairs. So you can see the first bit of data just here is our first aptamer binding onto the sensor surface, capturing the target protein. So aptamer at the bottom, protein sticking to that here, and then a second aptamer going over the top. So obviously if that second aptamer is able to bind, it must be binding somewhere else on that target. If it was binding to the same site, you would expect this to either be flat, so it isn't able to respond, or it to start knocking some of that target off. So this is exactly how we're going to be screening the aptamers that we've developed here, using that sort of process. Now obviously, biosensors like that, very big bulky machines, it's not something that's easily portable, something you can do at home. So you need something that people are used to doing and is easy to do at home. Well, everyone got used to doing COVID tests, everyone hated them, but they work. Okay? So like many people, when COVID hit, we had a choice. Shut everything down and send everyone home, or work on COVID. What do you think we did? We worked on COVID. So what we were able to do with that is take that COVID spike protein and develop a pair of aptamers. One that recognizes the bottom of the spike and one that recognizes the top. Okay? And again, very, very quick. We did that in 17 days. Okay? We got a pair of binders against the COVID spike in 17 days. And we were able to show that that worked really, really nicely in an ELISA format. That's great, start point for a lot of tests. We showed with a collaborator up in Glasgow that we could put that onto a biosensor and the aptamers would recognize COVID in clinical samples. Very, very clear result there, showing the positive response on the far side and negative result on this side. What we were also able to do was demonstrate that we could put this into a lateral flow. So hopefully you can see where we're going with this. So you can see very nicely a concentration-dependent response in the lateral flow test, detecting the COVID virus. Okay? And one thing that we were really excited about, this was a result we weren't expecting, but it was great. The aptamer recognized, or the aptamer pair, sorry, recognized every variant of concern that we tested it on. Okay? So wherever these aptamers are binding, it's not affected by the mutations that were arising. So that was a great result. So hopefully, what you can see is a story developing here. We've got a T14 target, we've got a selection platform, develop a pair of binders that will go into a lateral flow. So the end goal here is to develop a nasal test that will be able to pick up T14, show that very early in the life cycle, and hopefully then be an indicator that you need to start treatment. So I hate reading summary slides, so I'm not going to do it, but I'll leave you with that, and thank you again for inviting me.
Okay. Right.
Nice.
Very good. They're on. Okay, and now we move on to the slightly more proactive part of today, which is the Q&A session. And again, it's Q&A in name only. Feel free to share thoughts, ideas, and also satisfy your own interest in what it is that we do at Neuro-Bio. It's a fully collaborative point. So any questions, first and foremost, to... yes, shock. Let's go from the right to the left with some questions to put on. Thank you.
Hi, my name is Rhonda Siddle. I'm a freelance writer, and this is a question to Baroness Greenfield. Given the inherence of the degenerative process before symptoms start, do you envisage nationwide population-based screening programs?
Yeah, okay. Eventually, yes. I think that if we can get a non-medically supervised go, no-go screen, we'd have to be very careful how that was presented. I don't think we'd want to say, test to see if you might be getting Alzheimer's or not. I think that its first use anyway would be perhaps for recruitment in drug trials, for enabling appropriate subjects rather than people that were totally healthy. You'd have people that could benefit. But eventually, yes, it would be something, you have to have something that's nationwide that doesn't require medical supervision. If you're going to screen people at the pre-symptomatic stage, you have to have that. But it would be very carefully presented. It would have to be just a go, no-go. Do you need to go and see the doctor? Or is yours the normal range? But I would certainly feel worried. It's a bit like with HIV or something like that. You wouldn't have that over the counter. But yeah, it has to be something that's nationwide. It has to be self-administered. Otherwise, you're not going to be able to screen the people in that magic 10 to 20 years.
And just a quick supplementary to that, you talked about FDA submission. How, I know you can't repeat it before, but how long might you say the timeline between FDA submission and clinical trial?
I'm going to turn to my colleague to the right, who's an expert on biomarkers and familiar with the FDA.
Well, it depends on two things, and that's the data we have to convince them, and the medical need. And there's no doubt that Alzheimer is a huge medical need, as I mentioned. And I think that the FDA has different pathways. They have prioritized reviews. So they certainly will do it the fastest possible. But you need, and the problem with Alzheimer is that the studies have a certain duration because you have to see the deterioration of the cognitive functions, and that takes months. So you always have to count that in. It's not like we were just discussing before, like a cancer, where you can see it shrinking in a few weeks. All of these things, but certainly the priority will be very high to get this validated and approved by the FDA.
Yes, so we'll go to the gentleman to the left, and then we'll go to Clive and then Dom.
Hi. So, apologies if I say something that was covered in the first hour, because I wasn't able to be here for the first hour. I'm a medical doctor, Raj. Can I just start? Thank you for the presentation. So can I ask David about your nucleic acid aptamers? Do they cross the blood-brain barrier? And if you attach a molecule, will it cross? Because obviously there's some leaky areas of the blood-brain barrier which are a bit more therapeutically accessible.
Yeah, it's a very good question. I hate the answer, but it's sort of a yes-no answer. So on their own, intrinsically, they don't cross the blood-brain barrier, but we know from some very clear work that's been done with this, you can actually get aptamers to shuttle across the blood-brain barrier. So you can actually piggyback on other molecules that already cross the blood-brain barrier, and they will take aptamers with it. So there's quite a lot of work has been done with aptamers delivering across the brain to do that. And they have been conjugated to other drug molecules and take them across.
So, and just to follow up, on some certain areas of the brain are more accessible. For example, if you go via the olfactory neurons, you have direct access to the brain, particularly if you get something to uptake in the neurons, using wheat germ agglutinin or horseradish peroxidase or something like that. Is there a technique of using these binders to use neuronal pathways in? What I'm thinking of is if you can target an area, because the aptamer eventually binds to the area of the brain that is developing a pathology, do you have to, will you have to use intrathecal methods to get this in the end, or can you do it either by an intravenous or intranasal route?
It's a very good question. I would expect you probably could do it through either intranasal or IV. So the work that I've seen, I think most of their injections, most of their administrations were intravenous, and it's circulated and then crossed. So I think you could do both, yeah.
Can I just say,
Nope,
yeah, in relation to Alzheimer's in particular, of course, another application of the aptamer could be as an agent, as a therapeutic, not as a marker. And given, I think it was Neus mentioned, or Paul mentioned, there's a breakdown in the permeability of the blood-brain barrier with Alzheimer's, that actually could be a nice way of regulating how much, the worse you were, the more got in, yeah? So, just
Thank you.
Yeah.
So we'll go to slide next.
Thank you, David. Yes, those preceding points are pretty much ones I was going to mention, but just to take it a little bit further forwards, is it conceivable that an aptamer could be designed, one that crosses the blood-brain barrier, and that could actually prevent T14's binding to the alpha-7 receptor? In other words, act as a sort of antagonist.
It's entirely possible, yes. So, we know from other work and other projects you can use aptamers to block protein-protein interactions. So yes, it's entirely possible that we could essentially use the aptamers almost as a mop. Yeah, so it's possible.
And one thing that we can try as soon as the aptamer group let us have some, that's on our list to try. Yeah, in the lab.
Absolutely. Thank you. So over to Dom next, please.
Thanks very much for your very interesting presentations. My question's on the sort of clinical side of things. I can see the academic interest in understanding the early progression of the disease, pre-symptomatic. And I'm just wondering whether you have any thoughts of how patients might be interested in this in terms of adopting it in a more routine, non-clinical, as Susan said, but environment. So whether people really would be interested to know about the progression of a disease that really could be life-changing for them and perhaps not very well treated, especially in the current lack of any substantial or substantive treatments.
Thank you for your question. It's very interesting and timely. Yes, I think that we foresee a future in which we will be able to diagnose the disease before the symptoms happen, and then we will be able to administer some treatment. That's the scenario that makes more sense. But right now, if we are detecting at-risk Alzheimer's disease, that will be useful, as Dr. Greenfield has mentioned it before, for clinical trials. And in general, the more and more we learn about the disease, and also the population is more informed about how this process goes, they want to know at very early stages if they are going to develop or not. We are frequently asked about genetic risk, for instance, and we have to explain a lot about ApoE and how we differentiate from the mutation carriers, for instance. So I believe there is an interest, and many people would be happy to test themselves if they see they can get a potential benefit with a drug trial that potentially can avoid for them to develop the symptoms. I would say so. Of course, it's very personal, and not everybody will be aware of what is going on, but a lot of them, I think, would.
Maybe as an addition to that, as I mentioned, we will see a much larger proportion of patients in the general population as we move on. So people will have started to see what Alzheimer means, and they will be probably much more motivated, provided there is a treatment at the horizon. I mean, if you look at the Huntington case, it's very interesting. Huntington, you can diagnose very precisely by the number of repeats if you're going to have the symptoms or not. But half of the people who have a parent with Huntington's don't want to know. Even so, they have a 50% chance of getting it. But that's mostly because there is no treatment. So I think the link between a treatment and the marker is crucial.
Absolutely, and I think just before we move on to Professor Johnson shortly, just to add my 2 pennies in this, it's very much about talking about those 2 stages of validation of that biomarker. Firstly, we have to peg it to something that we can see. We know that neurodegeneration and the cognitive ability will start deteriorating, and as soon as we start pegging our biomarker to those symptoms, we can then really start moving away as a clinical stage company ourselves, but also helping other clinical trials, once we actually establish that link, move away from.
I should say that we're thinking of 2 stages, the go, no-go home test and then the medically supervised, more quantitative test. Yeah, but in the go, no-go, it would simply be to tell you to go to the doctor, that's all. It wouldn't be telling you anything else, just say, like you might have, it wouldn't necessarily be a positive diagnosis, just tell you need to get further checking, which is, if you couch it like that, it doesn't sound like having some terrible definitive sentence passed on you. Yeah.
Absolutely, but it does help us build that foundation of the data. The more people that will take this test, the more people will then have providing that kind of link to any further clinical stages of that disease moving forward. And yes, it will be a deeply personal decision as to whether or not that individual chooses to take the test. But I think there'll certainly be enough for us to start building that data and that link.
And I think also, I've been talking with Paul about this, it'd be nice to, the validation simply be just in the controls at the moment. We just want to establish how robust that metric is, because it seems very tight. And if we can establish there's a robust normal metric, that's obviously from which everything else deviates, then that would be a great step.
Exactly. So over to Professor Stein.
Thank you. I've listened to this with great excitement, but my question is really directed at Susan and Paul. I've always asked you, Susan, about this switch of the T14 evil property, or to an evil property, from its role in development. How do you think that switch occurs? And the bit that I'd like Paul to answer is whether or not he thinks that switch is likely to play a part in his speculation, as he says, about whether or not it could play a part in the pruning system that goes on early in the infant's brain development.
Okay, so I think we shouldn't really call it a switch, because that implies that it's a quick, on-off digital. It's more a spectrum of. Okay, trophic to toxic. Okay, the toxicity arises, and this is well known. If you have too much calcium going in and it's not appropriately mobilized, it is taken up into the mitochondria, where it compromises oxidative phosphorylation. You therefore get a leakage of electrons, formation of free radicals, and destabilization of the membrane. So that mechanism is a known one. The question is, what determines whether the calcium is going to be put to good use or not in terms of the age, in terms of promoting the neurite growth? That, I think, is a spectrum. The other complication, which means I can't answer specifically that X happens and suddenly it becomes evil, is we are currently working, and this is very exciting, on 2 stages in Alzheimer's. We think the initial process, as I described, which is an intercellular, where you have the T14 as a signaling molecule binding to an extracellularly expressed receptor that then triggers the calcium. But our current work, without giving away too much or too much detail, suggests that once that happens, what could then be triggered in the cell is something at the level of the gene, with something called alternative splicing, whereby the evil T14 is produced inside the cell, on its own. And that, again, presents as another therapeutic target, one that's way down the line, where we would intervene at the level of the cell nucleus. So I don't think we should expect it to be a switch. It's not like everything in life, it's not simple. I think that certainly what might make the difference is that switch, if you want to call it, that transition from being an extracellular signaling molecule, which is what happens, I would suggest, in the early stages, to then converting that into the erstwhile developmental mechanism, which is a genetically programmed mechanism, which would kind of make more sense, because if it's aberrant development, that's in lockstep with day for day for day. You don't want it haphazardly, relating to proteolysis in the extracellular space. So I think that it's an interesting question. I don't think it's something that is a a deal breaker for the theory. I think it just suggests that we do need to explore much more fully the biosynthetic pathway of T14 because it's more interesting than we had initially thought. And it does involve, although I've always steered away from this, actually looking at the level of the gene.
Yeah, I'm fascinated by it. Paul?
Yeah, Paul.
Yeah, maybe, so in my speculation, T14 would always destroy neurons. But there's an appropriate time for that, which is during the pruning process. And in the wrong circumstances, it becomes pathological because it still destroys neurons, but you need them. So the switch is rather... No. There's pure. I don't have a lab anymore, but we can. But what we could.
But there is evidence that T14 can increase outgrowth of presumably useful synapses or useful.
Yeah.
That would be great in development.
That fits your theory.
Can I say that another if we're going to wild speculation now, because we're among friends, yeah.
We're among friends.
Well, we're among friends, okay. So what I find interesting is in a control situation, the intimate relationship between phosphorylated tau and T14, which is very tight, and something we've been speculating on the healthy normal. Perhaps that's a mechanism preventing the T14 being toxic. Yeah, it's a natural
or the switch
possibly
might be stop producing T14 after early development.
In the intracellular mechanism, yeah.
And then for some wrong reason it starts again later.
Well, it starts again because it thinks it has to grow again and mobilizes things, yeah.
Fun discussing that.
He's called Professor Ralph Carter.
Hi, thanks. Sorry, is this on? And it's great to hear that you've now got your hands on some useful T14, and it's going to be fascinating to see how this is shown to be of use in the clinic. And just to ask, do you expect T14, apart from being a diagnostic tool, to be a clinical monitoring tool? And then looking forwards further, when it comes to the registration studies, will you be hoping FDA and EMA to use a biomarker endpoint, or will you be looking only at the clinical endpoint, which, if you're looking at preclinical prodromal disease, to be decades?
Well, it depends what a potential drug would do. Because if you simply have an antagonist of T14, unless you change the level that goes in parallel with the level of T14, if that is the case, then you can, of course, monitor the disease. Because T14 disappears again, well, it cannot be toxic anymore. So then you could contemplate what you just said. If it's only competitive antagonist that prevents the effect of T14, but does not affect the actual concentration of T14, then you couldn't do that. This will have to be tried out.
Person else wants to. Do you want to comment on that?
Well, I think that sometimes, as Dr. Herrling said, depending on how the mechanism works with this molecule, it might be a good monitoring biomarker or not, but not always all the biomarkers that we use for diagnosis are useful to monitor prognosis or to monitor the disease course. So it's something that we should figure it out as we advance in the process, I believe.
I think just to add to that as well, there are two puzzle pieces that are worth mentioning, which is number one, again, another benefit of us having the diagnostic LFT aspect here is the fact that it starts building the scientific validation of the theory of the connection between the two. So hopefully by the time that we get to the efficacy clinical studies, there'll already be some acceptance, not just from regulators, but the wider scientific space that there is that connection if we have the data to prove that. And secondly, the timelines that you're describing are very much the reason why we're so open to collaboration with other companies in this space for the biomarker, just so the kind of budgets that we're talking about in order to get this kind of biomarker over the line in terms of validation are so large that it will take us, of course, longer to do that ourselves. Then we'll find a partner with the right budget and the right sort of appetite to help us do that, to increase the speed at which we can get there. And hopefully, again, by the time that we get to the efficacy studies of phase 2 onwards, there'll already hopefully be, again, that link that a regulator will accept as potentially enough. But it's a very much work in progress.
Can I also add to that? I think since we had a party last year at the House, it was in April, and I think since then the company has really changed and its prospects have changed. Now that we can collaborate with hospitals such as Neus has and with companies such as Aptima, it really is giving us traction. And I really feel now we are moving much faster than we did even a year ago. And I think there's a realistic possibility, given if this metric holds, if the Aptima work is going as it's going, we've identified a lateral flow company already who promised to deliver a kit within 3 to 6 months, they said. That's what they said. Well, so you could work out, we could have, let's say, a kit ready at the end of the year. No, it would have to be validated, tested, blah, 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 but we would have physically something that we could see what its potential was. Yeah.
And that's a huge benefit. That's a huge benefit of exactly what our mantra is now, which is that collaboration, realizing that there are specialists in different fields, from LFT to Aptimas to small molecules, and really leveraging all of the appetite that actually, as a field of medicine, we have. I mean, we know this is a huge area of clinical problem that is not going to go away. In fact, it's going to be growing especially in geographies which are just unfortunately least prepared to shoulder the burden that Alzheimer's disease produces. I mean, to wider societies, right now we had a link between older populations and wealth in countries. And wealthy countries and wealthy populations are best positioned to shoulder that burden of the Alzheimer's disease space. But unfortunately, as the populations age in, say, Africa and the continent of Africa, we're going to be seeing the same progress of the disease prevalence in that population. And they are less prepared in terms of economics to shoulder that burden. I think that's the real strength of what we're preparing here, is especially on the Aptima side, because of the fact that the logistics are so much more simple. Simple means cheaper in this space, that they're inherently linked. So I think being able to provide something that will not only be pigeonholed into populations that can only afford it, but more widely, I think is another huge benefit of what we're working on.
Especially if, and this is huge, wild among friends speculation, if the Aptima also proves to have a therapeutic application, to be able to ship something to countries where you didn't have to rely on temperature, and it was light and it was powder, then that would be a real game changer. Yeah.
Okay. Any more questions? Yes, very good. So we'll start with Margaret and then we'll move on.
Thank you. I'm Margaret Aziri. I'm a neuropathologist. I'm still a bit puzzled by how the Aptima works. How does the nucleic acid have the specificity that's required for this particular peptide? What's the basis for it? It's not related to the nucleic acid that codes for that protein, is it?
No. So essentially, you can think of the way I always like to describe Aptimas is almost like one half of a piece of Velcro. So if you take a strip of Velcro and peel away one side, you've essentially got a ribbon with a whole load of hooks that are free to do something else. Now, all of those hooks in normal DNA are involved in holding the two strands together. But when you've only got one strand, as all of our Aptimas are, you have all of those hooks are essentially available then to start interacting with other things, like the side chains on the proteins on the peptide. So it's not that the Aptima codes for the protein, but it's able to use those the bases of the nucleic acid to interact with the protein itself. So it's basically a network of interactions as the Aptima wraps around the target.
But is it related to the sequence in the DNA that codes for the protein? How do you match up the sequence with the peptide?
Yeah, so what the Aptimas will be doing, again, as they're single-stranded, some of those single-stranded regions will be involved in intermolecule base pairing. So they will help the Aptima form a particular shape, and that shape will expose other bases that are then able to interact with the target. So there's two things going on. Some of the molecule is involved in shape, some of it is involved in interaction with the target. And it's that combination of the two effects that allow an Aptima to interact with almost any sort of target.
Thank you.
Okay.
Very good. Now we'll move to the back.
So I have a question for Neus. So since the locus coeruleus is the first to be destroyed, and we know locus coeruleus is wake promoting, so do they suffer from excessive daytime sleepiness at the beginning? Because we know after they have more time spent awake.
Yeah, when there are different studies, and in general, what we see in Alzheimer's disease from early stages or even in older population who has changes in AD biomarkers are nighttime awakenings, but also daytime sleepiness in comparison to healthy cognitive individuals, and also they have an opposite pattern from other tauopathies or other diseases such as PSP, as I mentioned it earlier. So we believe that the degeneration of LC is driving some of these changes, but of course, not all the changes, all the clinical symptoms are driven by locus coeruleus, but probably by other IC neurons within the brainstem, not only LC. However, we are focusing on LC a lot because it's one of the few that we can measure in vivo by MRI. Other nuclei, since they are so tiny, they don't have neuromelanin, you cannot measure them accurately, at least.
Fantastic. Any more questions from the audience? Very good. Over to David.
Thank you. David, Neuro-Bio. Thank you for 5 great presentations. I've got a question that sort of follows on from that question. So if you have a treatment, MBP14, would that have the possibility of managing the sleep disturbances, the depression, the constipation, all of those things? So you wouldn't have to take treatments for that as well as MBP14?
Hopefully, that would be a great aim to, if we prevent the neurodegeneration to start within the brainstem, we should avoid the sleep or the depression symptoms if it's administered early enough, before all of them start.
So I've got a question therefore for Anton. Does MBP14 IP cover depression? Could it be an antidepressant?
The broadest we go is neurodegeneration. So if depression is defined as neurodegeneration, then yes. If no, then no.
Exactly.
But it might be preferable to, yeah. Just think of the market there. We go from
And treatments for insomnia.
Yeah.
Indication from neurodegeneration. Suggested probably is, then it could be protectable independently as an alternative medical.
Wow. Well.
Another therapy.
So that'd be number 21 of our pattern families.
Hopefully everyone is bound by confidentiality in this room.
Very good. Any more questions from the room?
I'd like to make one
Fantastic.
more comment.
Am I allowed to ask another?
Of course you
then
are.
I'm asking. Yeah.
Extremely left field, I'm afraid. I'm interested in nutrition, and I know that Alzheimer's has, it has been suggested that certain nutrients, or the lack of them, is very important for Alzheimer's, and indeed there's quite a lot of suggestive evidence that that's the case. In particular, omega-3 polyunsaturated fatty acids, vitamin D, and a few other molecules. Any thoughts from anybody on that?
What's the question?
The question is, how does nutrition play into your theory? Sorry.
I cannot.
Sarah.
I think we know that choline can be derived from the diet, and we know that choline activates the receptor. That is where T14 sits and lets all this calcium go in. So perhaps things related to choline wouldn't be recommendable in an Alzheimer's situation.
Yeah, also we know there has been, I'm rusty on the literature, but I know that Alzheimer's has been linked to problems with choline uptake mechanisms. And choline, as Sarah says, is the primary ligand of the alpha-7 alternative to acetylcholine. And it's derived from the diet, so that's good. Oh,
One more question.
Obviously, in medicine, we'd love to have the idea that there is a single common pathway which will cure a disease. But most of the time it isn't like that, and there's multiple different pathways. So when you look at Alzheimer incidence, and my colleague here mentioned the nutritional aspects. For example, we know that cinnamon workers have a very low incidence of Parkinson's disease. People in epidemiological studies suggest curcumin may be helpful in slowing down or perhaps prevention of Alzheimer's. And then we know that Alzheimer incidence rises, as in the clinical state of Alzheimer, with, for example, type 1, type 2 diabetes, and also hypertension. So what I'm asking is do you think, and going on with some of the other questions, do you think your process is like a final common pathway, or there are going to be other pathways anyway that lead to Alzheimer's? I mean, if you hit this, do we still need to control the others optimally?
Yeah, I think obviously everything is a sort of old adage during neuroscience, everything's connected to everything else and everything causes everything else and so on. I think that the process that we are working on is very upstream, and it is a very basic, simple process in biology for promoting cell growth. I'll come on back to that in a second. I think because it's so far upstream, it will then cause a concatenation of events that, depending on the neuronal populations in which that is occurring, will then trigger different things like ApoE or inflammation and so on. But thank you for that, because what I was going to say to the chairman, I'd like to widen out, because we're very privileged to have two people from Genepat in Barcelona who've been collaborating with us on cancer. And if I may, Raj, I'd like to build on your question to say that I think this process is so basic, not only does it trigger whole concatenation in the brain, but it also could be irrelevant to other systems outside of the brain where you have inappropriate cell growth or renewal, which is, of course, a definition of cancer. So with Chairman's permission, perhaps I wonder whether any representative from Genepat would like to just say one or two words about what you do and how this is relevant to your process in oncology.
Hi, I'm Anna from Xenopat, and yes, we've have the pleasure to be cooperating with Neuro-Bio in testing NBP14 in different cancer cells. Especially, we're working in colorectal cancer, and we have preliminary results that show reduction of the tumors when the animals are treated. So, as Susan explained, maybe the initial collaboration came up since they checked the expression of T14 in those cells, and it was upregulated. So that was the idea to try to treat them. And it's working. It's preliminary data. We have to go further, but we're working on it right now.
Fantastic. Okay. Any more questions from the room? And perhaps any on the oncology space?
Sorry. And having disclosed that I come from oncology, so not a specialist on neurodegeneration, so what about resistance? So if we're planning to start treating the patients as early as possible, because we're going to be able to detect the disease before they have the symptoms, we have to treat them for years, hopefully. Are we expecting them to become resistant to the treatment?
Maybe, from cancer, of course, very well that all the attacks that we devise become resistant over a while. So I think the answer to that might be a panel of antagonists. So when you get resistance in one, what you also do in cancer, you take another one, hoping that it's a different site, and you can restore the efficacy. But of course, that's still early. So we'll have to do the experiments, but at least there are precedents how you can get around that. The same is in malaria, for instance, and these kinds of disease where resistance is an absolutely huge problem.
Just to carry on, for the MBP binding to the receptor and displacing T14, I can't simply see, I don't see how that could desensitize, because it's still occupying the receptor site. It's been rendered inert. And so it's not as if it's it's not like L-DOPA in Parkinson's, for example. It's not as if, or indeed antidepressants, where you do get resistance to the drug or receptor-mediated changes. The fact that it's occupying the site and having no effect in and of itself, and just preventing the toxin getting there, I can't see how that would change, how that could desensitize or develop a resistance. Personally, I don't see it unless I'm missing something.
I think that's one of the theoretical strengths here, is the fact that any potential mutation in the receptor for T14, which would render NBP14 unable to bind to it, would then in its own turn make T14 be unable to bind to it itself. So that sort of resistance pathway, I think, would be relatively difficult to, because they bind in exactly the same way.
Yeah, NBP does nothing, and it just sits on an allosteric site, so there's nothing, nothing to change. Yeah.
Anton, I think you had a.
It's related actually. So back to aptamers again. I mean, NBP14 is 14 amino acids long. It's cyclic, so it's quite a relatively not as big as an antibody, but it's a big thing. Aptamers are significantly orders of magnitude smaller than NBP14, so we know that the aptamer will be binding to T14 somewhere, so it can be used to detect in LFTs and all of that. That's great. Therapeutically, how would such a tiny single strand of DNA, 60, 70 nucleotides long, how would that potentially work therapeutically? Would it also sit in the same allosteric site as the cyclic peptide, or would it stick to T14 at an angle and prevent? Because the sizes are so radically different, I can't understand, and I don't know the three-dimensional structure of the allosteric site. Do you see my sort of problem here? There's a disconnect between a big cyclic peptide and a tiny DNA molecule.
Yeah, true. I mean, obviously the answer is very theoretical. There's quite a few ways that an aptamer could inhibit a protein's function. So some work that I did a very long time ago, I generated an aptamer that recognized a small loop on the outside of that would have been about a 15 kilodalton protein, I think it was, and just recognizing that small loop that bulged out of the side of a beta barrel was sufficient to stop that protein unfolding and going on to form an amyloid. So you don't really need to have a big molecule that encompasses the whole protein to have quite a dramatic effect. So, just because it's a lot smaller doesn't mean it wouldn't be effective.
And as we know from our exploration with different variants of NBP14, exploring the binding of T14 to the receptor, it's a very precise situation. You wouldn't require much, I would imagine, to intercept or interfere with that. Size isn't everything, Anton.
So the aptamer's sticking to T14, which would prevent it from slotting in, rather than what NBP14 does, which is it doesn't touch T14, but it blocks. So it's the lock and key. Yeah. So the aptamer is sticking to the key, preventing it from hitting the lock. Yeah. NBP14 blocks the lock.
Yes.
Exactly.
Yes.
I think at that point it would be a balancing act between, again, we will know, for example, that NBP14 works through effective competition of that binding site, but it has, it's still a protein. It's still relatively difficult in terms of logistics to administer. We have the easier potential, theoretically, aptamer to do that, but as we're talking about just resistance, if we have that aptamer either deforming somehow the binding site of the receptor itself or deforming the T14 as a molecule itself, perhaps we'll have to have 2 or 3 options, as we were talking about, in terms of that resistance, those mutations, perhaps in making that one single aptamer work slightly less well, less effectively.
You wouldn't need to conjugate it to any kind of payload necessarily. It's not like a cytotoxic agent where you want to kill the cell. You just want to, maybe longer aptamers would improve the blocking capability.
Yeah, you just want to bend the key. Yeah. But can I ask, David, when can we have some? Because we can test this easy. I mean, the team are here. We're dying to get some. So weeks, months, days?
17.
Tomorrow? Yeah.
It certainly won't be tomorrow. I would guess it will be within a month or 2.
Yeah. Okay. That'll be exciting. So we'll have to reconvene in a month and find out.
See you all here then.
Yeah.
Any more questions from the room? Now, because I have just one very quick one for Neus in terms of the clinical sort of pull and need for this. From your perspective, you must see, by the time that patients get to you, as you were saying, they already will have been prescribed the antidepressants, the sleeping disorder medication. I imagine it's relatively frustrating for you that we as a field missed those early stages where, as we now know, it would be the most effective to start the treatment. Ultimately, brains are very good at filling in the gaps that form through neurodegeneration. So by the time that we see those cognitive impairments, it's a little bit too late. How is it from a clinician's point of view to deal with that, with the so far the shortcomings of our ability to deal with the problem?
As you say, it's a little bit frustrating because when most of the patients, they have been prescribed with antidepressant medications, and sometimes they are not effective because neurodegeneration is the cause. It's not because a psychological problem or a primary depression syndrome, but it's related to neurodegenerations, and you don't have a lot of tools to try to deal with these symptoms. And then memory problems that, of course, you have also very limited strategies. So you can offer limited treatments to everything and try to inform patients and family about the prognosis, but unfortunately, it's not much that you can do right now to improve their lives, but providing care and helping them on the process. Yeah, sadly.
Absolutely. And then one last one, I guess, for Paul, and this is from a commercial point of view, from your time and expertise from Novartis' side, etc. This kind of biomarker would potentially be absolutely transformative. But as we know, human nature is sometimes to potentially hide our heads in the snail's shell when we find something that is potentially so disruptive. Do you think Neuro-Bio sort of technology now, or the therapeutic, we're seeing perhaps more of the hiding the head aspect. On the biomarker, do you think that might be slightly different? Do you think there'll be a huge appetite from the commercial arm of the medicine, or not so much?
Well, I'm not sure. A pharma company typically would only be very interested if there was a companion drug. Yeah. And you could use that biomarker to speed up or make the clinical studies cheaper. But a company like Roche, who has a very big diagnostic department, might be extremely keen to add that to the portfolio. So if I had to choose, and not being a Novartis previous person, I would go to Roche first. Nowadays.
Yeah. This is off the record.
Yes, this is very
No,
much
no, I'm free, I'm retired, I can say whatever I want.
Very good.
Do you have friends in Roche?
Find some.
Yeah.
You know, the ones that were Novartis before.
Oh, okay.
Fantastic. In that case, we'll now end. We've still got a little bit more time, so I very much invite all of you to find the panelists, knock some more ideas around. But very much thank you all for coming. Really, really appreciate it. And huge thanks to our amazing panel. Really appreciate also keeping to time on everyone's speeches. So huge thank you from me and the wider Neuro-Bio team. So thanks all.
Thanks to you as chair.
And I will see you shortly when we do some mingling. Thank you.